Bismillahirrahmanirrahim. Assalamu alaikum. My name is Fatima from DS Biotechnology, and today my topic of presentation is nucleic acid. Nucleic acids are the biopolymers or large biomolecules essential to our form of life. The term nucleic acid is the overall name of DNA and RNA, deoxyribonucleotide. They are composed of nucleotides, which are the monomers made of three components: a 5-carbon sugar, a phosphate group, and a nitrogenous base. Here, this is a structure of deoxyribonucleic acid, means DNA. Here we can see sugar phosphate backbone. These are the base pair of DNA. And this is thymine, guanine, adenine, and cytosine. And this is ribonucleic acid, RNA. This base pair is adenine, uracil, guanine, and cytosine. The difference between their base pair is that thymine is absent in RNA and uracil is present. DNA and RNA. Deoxyribonucleic acid and ribonucleic acid are molecules that carry genetic information in cells. DNA is the molecular archive of instructions for protein synthesis. RNA molecule transcribe and translate the information from DNA for the mechanism of protein synthesis, means the synthesis of amino acids. The storage of genetic information, its passage from generation to generation, means the heredity, and the use of genetic information to create the working parts of the cell all depend on the molecular structure of DNA and RNA. These are the nucleosides of DNA. There are 4 nucleosides present in DNA: base pair, adenine, guanine, cytosine, and thymine. These are the nucleoside of RNA. Here, thymine is absent and uracil is present. And this is a basic difference between RNA and DNA. Synthesis of nucleotides. Synthesis of nucleotides means synthesis of their synthesis and their degradation. Mild degradation of nucleic acids yield monomeric units called nucleotides. A general formula for a nucleotide and a specific structure of one called adenyl cyclic acid. Complete hydrolysis of a nucleotide furnishes a heterocyclic base from either the purine or pyrimidine family, a 5-carbon monosaccharide that is either D-ribose or deoxyribose, a phosphate ion. These are the 3 things that we got when a nucleotide is hydrolyzed. This is a nucleotide. Here it have heterocyclic base, nitrogen, and this is a adenyl. Synthesis of nucleotide. Removal of the phosphate group of a nucleotide converts it to a compound known as a nucleoside that can be obtained from DNA. All contain 2-deoxyribose as their sugar component and one of 4 heterocyclic bases: adenine, guanine, cytosine, and thymine. These are the base pair of DNA. The sugars present in DNA are RNA. DNA and RNA are distinguished on the basis of pentose sugar present. DNA contains beta D2 deoxyribose and RNA contains beta D ribose. Here, these are the structure. Here we can see that this is a hydroxyl group, and here no oxygen is bonded to this carbon with the hydrogen. Metabolism of nucleotides. Here we can see metabolism means synthesis of nucleotide and then their degradation. Here we can see a molecule alpha D ribose 5-phosphate will convert into 5-phosphoribosyl alpha pyrophosphate. In short form, we can say PRPP. Here the ATP is converted into AMP. Adenosine triphosphate is converted into adenosine monophosphate with the help of PRPP synthase. This is the enzyme which converts RFP into PRPP. Then PRPP is converted into PRA, beta 5 phosphoribose ribosyl amine with the help of PRPP glutamyl aminotransferase enzyme. Here the glutamine is converted into glutamate and what with the in the presence of water and PPI. Here the PRA is converted into glycinamide ribotide GAR with the help of GAR synthetase enzyme. Here the glycine is converted into PI and ATP is converted into ADP adenosine diphosphate. Then the GAR is converted into FGAR farmiglyl cinamide ribotide, and the enzyme that is responsible for the conversion is GAR formyl transferase. Here NN is transferred into THF. This is methyl formyl. THF is methyl formyl. Then the FGAR is converted into FGAM, means formylglycinamide ribotide, with the help of an enzyme called FGAM synthetase. Here the ATP plus glutamine and water is converted into ATP glutamate and PI. Then the FGAM is converted into 5-aminoimidazole ribotide AIR with the help of AIR synthetase enzyme, and here the ATP is converted into ADP molecule, phosphorus and water. Then the AIR is converted into carboxyimidazole ribotide CAIR with the help of an enzyme called AIR carboxylase. Here the ATP and ASO3 molecules are converted into ADP and P. Then the CAIR molecule is converted into 5-aminoimidazole and succinyl carboxymide ribotide SAICAR, its short form, with the help of an enzyme called SAICAR synthetase. Here the aspartate is converted into PI and ATP is converted into ADP. Then here the SAICAR molecule is converted into 5-aminoimidazole 4-carboxymide D-ribotide AICAR, its short form, with the help of an enzyme adenosine lyase. And here fumarate a molecule is released. And here we can see fumarate is released. Then the AICAR is converted into 5 amidazole 4-carboxymide ribotide FAICAR. Here the THF formyl is converted into THF, and it is the enzyme used is AICAR formyl transferase. Next, the FAICAR is converted into inosine monophosphate, which is a final molecule compound, and it is converted with the help of a IMP cyclohydrolase enzyme. Then this IMP is further converted into AMP, and AMP is further converted for the production of energy. The biological importance, the function of nucleic acid have to do with the storage and expression of genetic information. Deoxyribonucleic acid DNA encodes the information the cells need to make proteins. A related type of nucleic acid called ribonucleic acid RNA comes in different molecular form that participate in protein synthesis. Xenonucleic acid ZNA are synthetic nucleic acid analogues that have a different sugar backbone than the natural nucleic acid. These synthetic nucleic acids are made in the laboratories for some purposes like therapeutic use. This is a glycol nucleic acid and is an example of xenonucleic acid, means synthetic nucleic acid, because it has a different backbone than the DNA, because DNA has a different backbone than this. One theory of xenonucleic acid utilization is its incorporation into medicine as a disease fighting agent. Xenonucleic acid also has potential application to be used as catalyst, like an enzyme catalyst, much like RNA has the ability to be used as an enzyme. Because we know that RNA is already using as a therapeutic agent to fight some diseases, and they are under clinical trial. Thank you. That's all from my side.